who can join here and they can discuss among the topics. And yes, we have a Dr. Prakash Alami, sir,
Thank
uh,
you.
the expert, as well as really our main motto that to learn something from the different topic. And Prakash sir having a good knowledge of HR. I think I can also see that last week we had a I think continuous two discussion or continuous two topics on discussion. Like one is research empowering, how to write the research proposal, how to submit the research proposal and all. So continues last week we had also discussed with the this same topic, and earlier also the topic was PGPR, plant promoting rhizobacteria. Even though the topic was not suitable, I mean not area of sir. Of course, he is a food microbiologist. Am I right, sir?
Some
So
that
so, but really, he have a good knowledge of agriculture also. He have a good knowledge of environmental, biofuel, and all the things. So, yes, today's topic is MBSI. We are organized every week that I already discussed. So, today's topic is bacteriophage therapy, hope beyond antibiotics. So, I will not discuss about this bacteriophage because I think some research scholar are also here, and faculty members also know about the bacteria, the specialized virus to target the and destroy the harmful bacteria that we know, right? But role of antibiotics, right? जैसे कि हम लोग जानते हैं कि fast designing and clinical trials, like some deep hope against some drug resistant superbugs like MRSA, right? So sir is with us. So I think bacteriophage that is also against cancer. Am I right, Prakash sir?
yes.
And there are some bacteria like Acinetobacter baumannii, Cephalococcus aureus, they can form the biofilm formation. So we have a Prakash sir, and he will discuss about the fast therapy. Sir,
Hello. Hello.
Deshmukh sir, good evening, Deshmukh sir.
Good evening. Good evening. So today I will interfere a little. The reason is that
Please, sir.
I know
Yes.
some attendee today, and you might be knowing there is a one association in India which is Bacteriophage Therapy Association,
Oh, wonderful.
and this year the conference they are organizing in Patiala. I think the Dr. Tarun Anand. She is the organized Taruna Anand. She is organizing secretary. So I will request first Dr. Taruna Anand. You please give information regarding the National Conference on Bacteriophage Therapy. Which scientists are involved and what is the priority research in this regard? Dr. Taruna.
Thank you, sir. Thank you so much. And a hearty namaskar to Deshmukh sir and his team. And sir, I have heard a lot about you. Sir, basically, I this is the first meeting that I have been able to attend in this society. So I'm really thankful to you to provide me this opportunity to introduce our society here, sir. Sir, this is a Society for Bacteriophage Research and Therapy. And it works with a mandate of phages, bacteriophages exclusively, with a goal of an AMR and under One Health. So we are holding this seventh international conference this time, and it is going to be in Hisar. Sir, ours is Indian Council of Agricultural Research, ICAR, and ours is basically a National Research Center on Equines. And in that, there is a sub center which mandates for the collection of microbes. So we have a huge collection of bacteria, viruses, bacteriophages. The same way IMTech and other repositories work,
Very good.
we also work in the same manner. And
ఇలా
we have a
రాసేస్తే ఫుడ్ బాయ్
collection.
లాగా అమ్మ కొట్టదు కదా నిన్ను ఆ? ఆ?
So we have a collection of around 5,000 microbes at our place. And
One minute, ma'am. Ma'am, please, 1 minute.
ఏం
Two
రాసేస్తా?
few
Yes, sir.
members are not
Yes.
muted. I will ask them to mute yourself, otherwise I will remove them. I can remove as admin. Please mute everyone, I request. Yes, you can proceed.
Thank you, sir. So basically, we are a type culture collection, and because we are holding India's oldest bacteriophage repository, so this time we have been given this opportunity to conduct this conference, which is going to be an international conference. And many foreigners from very, very well renowned researchers from abroad are joining in that conference. And the main aim is to bring bacteriophage therapy as an alternative of antibiotics. So we have many researchers all over India who have gathered, joined together through this society. This society was established in 2017, and the first conference was held in BHU, Banaras Hindu University. And there the Ganga Phages were highlighted mainly because we know that Ganga water doesn't get rotten easily. Rather, we all have Ganga water in our homes, and it doesn't get rotten. So the thing that was highlighted since ancient times, that it is having bacteriophages. And so with that mandate, we proceeded further. And all over India, there are many groups who have joined together their hands through this Society for Bacteriophage Research and Therapy. So, sir, through your platform, I really invite each and everyone to please join this conference. They will be really benefited. Thank you once again for providing me this opportunity. Thank you, sir.
I will request you, give your contact number in the chat
Yes.
box.
Okay, sir. Okay.
So anyone who is interested
Yes, sir.
to attend the conference in Haryana, what is the date, ma'am?
Sir, it is 3 and 4th November. And
Okay.
there is a pre-conference workshop also, which is on 1st and 2nd November. And
Very good.
many students are already applying for it. And like we are thinking of selecting at least 20 people. So because this is a kind of new upcoming emerging area as far as AMR is concerned. So there is a pre-conference workshop on 1st and 2nd November, and there is a conference on 3rd and 4th November.
Very good. Now, within 2 to 3 minutes, you can explain your research on the bacteriophage.
Okay, sir. Thank you. Thank you for providing me this opportunity again. Sir, basically, we initiated this work in year 2013 because ours is a type culture collection. So we have a mandate that anybody who has microbes and they can deposit it without any fees, they can deposit it to our culture collection the way we deposit our money in bank. Similarly, any researcher can deposit their cultures so that they remain preserved for posterity. So they can deposit their cultures in our center without any cost. And any researcher or student or even private people also, they can get it by MOU, by signing an MOU, they can get those cultures. So I have like a few PhD students, master students, and we have carried out projects on. Initially, we carried out institutional projects, which were supported by ICAR. They were like development of the bacteriophage repository. So later on, we did on isolation of bacteriophages against mastitis causing pathogens. That was also a huge success. Then I got Lal Bahadur Shastri Award from ICAR, under which we got an opportunity to work on poultry pathogens. So all those poultry pathogens, which like Salmonella, E. coli, and Campylobacter, and all which cause infections in poultry, and they were isolated, they were characterized, and now also I'm working as a ICR National Fellow, and that poultry related work is being proceeded further. Within this time, we have published around 20 peer-reviewed papers on bacteriophages, and the one that is highest in impact factor is around 20.5. It I got it just last year, and we have worked on various aspects like how phage and antibiotics they can synergize, and what are the various characteristics which we should be taking care of while doing phage therapy. And we have established 3 animal model studies also, in which phage therapy has been applied, sir. We have done it in mouse model, we have done it in poultry, and we have done it for mastitis in poultry also. So it is matlab, I can say that fast with all those studies background, I'm able to highlight that phages are a good option for overcoming AMR. So I would request anybody who is in and around Haryana, they can come and visit, and I would really welcome that. Welcome all of you, sir. With this, I thank you once again.
Yes, madam. Not only nearby Haryana. I will
Yes, sir.
request right from Tamil Nadu, Kerala.
Yes, sir. Yes.
Students
Thank you. Thank you.
should visit.
Yes, sir. Yes, sir.
Very nice platform you have given to Indian bacteriophage researchers. So,
Sir.
today, more than 22 are attending, and I welcome all PhD holders. Again, I request if there is any PhD holder who is working on bacteriophage, please share your experience. Any researcher in bacteriophage. Because one PhD student has written that I am interested in clinical studies. So, any researcher have done the research in the area of bacteriophage therapy, please go ahead. Otherwise, Dr. Prakash Halami is there who can also focus the light. There is a
Good
Richa
evening, sir.
Gupta. There is
Yeah,
a
we can take some question from the PhD students
correct.
because they will
Kamal
be
Khan is
actually
there.
trying to work on this topic, and Dr.
Yes.
Taruna is there, so she will be right person to answer how
Yes,
we
yes.
can obtain the bacteriophage, how to check the specificity and selectivity, what are
Very correct.
the experiments are needed for a preclinical clinical trial animal model.
Yes.
And
So
how
now,
fuzz can be multiplied, stored in a larger amount, and all these things.
very good.
She has already the repository. So based on the question, I think we can go with that.
Yes, someone was asking the question. Please tell your name.
Dr.
Who is
Shiv
asking the
Sharma is
question?
also there.
Dr. Suresh is there. Dr. Thangmani is there. Dr. Kali Dose is there.
Dr. Shilpi, are you there?
Yes. Dr. Sunitha.
Shobna, Shobna
Nilima.
Raj having a question. Startup company Swastha Fire Solution. I mean,
Okay.
we are working on bacteriophage, sir.
Oh, wonderful.
Okay.
They can.
Very good.
They can
So she
interact.
should continue. Please.
It's better to interact those who has a practical experience.
Yeah.
Well, actually, very few people are working right now as PhD exclusively on bacteriophages, and there are many groups, although, and I would really welcome you, through your platform. I would really like to welcome people to work in this area. There is a lot like India can do because we have a enormous bacteriophage diversity in our environments, and they are quite easy to isolate, although the further studies are little bit tricky, and but it gives us a real hope to overcome AMR in coming future, sir. And
Definitely.
in a few months back, sir, I went to CFTRI Mysore also for a presentation as a speaker, sir. Dr. Halari sir was Hakari sir. He invited. So I went to CFT, not CFTRI. It was some institute, private institute, some university.
It must
So
be in Mysore
Mysore,
University.
in Mysore, yes, sir, yes,
Yes,
sir.
yes.
So that, but I would really like to welcome people if they have any query or any, they can contact me anytime. Rather, you can contact to anyone. Like we can kind of connect you to other researchers also who are working in bacteriophages.
Definitely, ma'am.
So
I
yes,
already
sir.
sent email to you. To your Gmail,
Uh,
you can reply. I am actually from CFTRI.
okay, sir.
There are actually one
So
or two questions, so I think that can be better. One good evening, Dr. Dipti Dhanwate from Mumbai, interested in FAS in clinical application. This is what our question is.
yes, sir. So, sir, clinical application like BHU has done some trials. Dr.
Uh-huh.
Gopalnath is there. He has done some trials, and rather he is a kind of a great person who has really dedicated his job, means his work towards this field, and he has really established some cocktails
Mhm.
in which the such phages are present, which can really help cure the means diabetes associated foot infections, and he has used them so as to there was no need of amputations, because in case of diabetes persons, the means wounds, they become very serious to heal. So he has developed some cocktails which have been able to save the lives of many people, and there are for if you are thinking of a clinical trial, the one thing that I would like to suggest is that phages must be pure. They should not be having endotoxins, and they should be completely sequenced, so that they should not be carrying any antibiotic resistant genes, or like integrases or recombinases. So means they should be safe to use and they should not be able to spread any resistance. So these two things are very, very critical if anybody has plans to go to clinical trials. So this would be my suggestion as of now.
Then there is one question about this
Arun, yeah, I think.
startup company, Swastha Phage Solution Private Limited. Shobna
Mhm.
Raj. Shobna Raj, you can switch on your mic and then you can ask directly if you have any questions.
Yes. Good evening, all. This is Dr. Shobana from SRM Institute of Science and Technology. And we have a startup company within the BioNest in SRM. And this is an initiative for phage therapy. Currently, we are focusing on bovine mastitis, and for poultry, we are preparing the phage product, and it's in initial stage. And I have a few PhD students working on three different fields. One is for Stenotropomonas maltophilia, which is a nosocomial pathogen. For that, we have identified phages. And then for bovine mastitis, especially for Staphylococcus aureus, we are concentrating on preparing a phage cocktail. And one more work we are doing along with the Central Institute of Brackish Aquaculture, SIBA, which is in Chennai. We are doing a collaborative work for finding out phages against pathogenic bacteria called Aeromonas hydrophila, which is a fish pathogen.
Wonderful.
Yes. So parallelly we are working, but we are in only 5%. We are in the starting stage in our startup company. So currently we are providing workshop related to phages for undergraduate, postgraduate, and PhD students. Those are interested in clinical department such as medical microbiology, or people who are interested in doing research work related to phages. They should know initially how to isolate, identify, characterize, and purify the phages. So currently we are providing internships, workshops, and FDP program for faculties. And along with that, product development, it is in preliminary stage now.
Wonderful.
Yes.
So basically, you need a formulation which will actually help to take care of Staphylococcus aureus associated with the mastitis or the Euromonas, the food pathogens.
Yes, sir.
And Dr. Taruna rightly pointed out that this phages should not have any, resistant genes,
Yes, sir.
the ability to integrate
Yes,
or
sir.
modify these genome structures of the host.
Yes, sir. We are focusing on doing the whole genome sequencing and finding out whether it contains antimicrobial genes or not, whether it is a lysogenic phage or not. All the
Exactly.
parameters we will thoroughly check, and then we will go to the production.
Wonderful. Dr. Narmada is there from Gulbarga University. I think she is working on the bacteriophage. I think we should ask her expertise or the work what she is doing.
Sir, Dr. Deepti here. My questions are...
Okay. Deepti can go ahead.
Yeah. So my questions are actually already answered by both of them. What we have found clinically, it is difficult. I'm a microbiologist, clinical microbiologist working in a hospital. It is very difficult for us to convince consultant that, like they mentioned that it should be free of endotoxin, there should not be any other enzymes, or there should not be any inhibitory substances. How do we gain their confidence in that? Because abroad they are using it. Here on compassionate ground, people have used it. But how do we convince our physicians to use them? That is one challenge. I mean, the workshop will help us. I got already come to know that there is a platform which is explaining. But how are we gaining their confidence that it does not have anything else which will cause more damage to the patient than help? How do we take care of that?
Actually, the thing is, after isolating the phages, there are a number of steps to purify the phages. against one particular bacteria, and production of bacteriophage, it's in preliminary stage in India as of according to me. There are certain companies which are producing, but with respect to endotoxins, that should be thoroughly checked, and it should be tried in vitro conditions, and then in vivo conditions, and then it has to go to patients.
I have a submission, a little submission. You have, madam, you have told very nicely.
Yes.
I have a little addition to it. There is one madam in PGI Chandigarh, Dr. Neelam.
Yes.
She is working on this aspect of treating patients with fast therapy,
Yes.
and means you can because this is a big challenge actually to convince the to convince the medico personnel how to use it. But one thing that can be added in this way is that phages, they are very very means sure shot in their action. They kill only their host, so they are not going to disturb any of the host flora, microbial flora, because they are so specific in their action. And the second thing is that I would really suggest you that you can talk to Madam Neelam. She is really working on treating the patients through phage therapy.
Yes.
And you can also contact Dr. Gopalnath sir, because he is also a medico, very senior dean. Then dean, he has remained for microbiology division. So he can also help, like how to convince, because there is some performa they can fill, and when you do not have any other option, so what is left with you? So then definitely this fast therapy provides a hope. It's like that.
Yes.
Thank you. Thank you.
Topical application, it's easy, and we can use phage therapy, for example, for treating Pseudomonas aeruginosa, for treating topical for veterinary cattle, we can use. Instead of inserting to get a regulatory confirmation, we can use topical application of phages instead of antibiotics to kill the microorganism.
But basically, like we have to line up, we have to develop confidence with the medico people. How and over time, this fast therapy is also improving.
Yes.
Like you can make a team of medico, and you can like really intervene by seeing whether really your fast therapy is working or not, whether some resistance is coming up or not, some new resistance is coming up or not. There are a few ways that are coming up nowadays. So, but
Please
definitely
don't disturb. Nice
convincing them is
discussion
a
is
big
going on.
challenge.
Who is there? Vivo, why Vivo V2315. Please unmute yourself. Nice discussion is going on. Otherwise, I will going to remove you. Vivo V2315.
म्हणलं मोठ्या खर्चाला आहे मी किरकोळ खर्च ऐकडनं काढत जा.
Ma'am, you may continue. Yes.
Sir, I have written my phone number and in the chat box, and my email also, in case someone has to contact some query or something. So maybe I'm not sure, but I can provide with my experience whatever I have regarding that, some queries if comes up.
Yes, yes.
Ji sir.
Ma'am, I'm from Gulbarga University, Department of Biotechnology. I'm pursuing PhD. Yes, ma'am. Is it audible, ma'am?
Yes, yes, please.
Yes, ma'am. Is it audible, ma'am?
Yes, it is audible. Yes.
Tapita from Department. I'm Tapita from Department of Biotechnology, Gulbarga University, Kalburgi. I'm doing my PhD under the guidance of Senior Professor Dr. Chandrakant Kelmani, sir. I'm actually working on FAS related to Acinetobacter baumannii. I'm actually performing all the experiment. I am getting some lysate, like I have got around 4 to 5 phases, but I want to go further for identification like whole genome sequence. And I need to know what all the parameters should be taken care that it should not go for the any resistant for the patient in future, and for any what is it, resistant mechanism or any other barrier to the patient. So what factors should be I take care that I can prevent these all?
For these, I told that genome sequencing is like one part that first of all we should be knowing that my phage is clear, my phage is clean, and it is not having any undesirable genes. That is one thing,
Yeah.
because it provides a mechanism. For example, it is having some gene which is homologous to some other gene that is residing in a bacteria. So, with that, homologous recombination can take place. So, in order to avoid that, that is the first way is that we should be going for genome sequencing. And the other thing is that we cannot resist, means we cannot prevent that because actually this evolution of phage and evolution of bacteria, they are parallel mechanisms. It cannot happen that if there is a phage given, then never ever resistance will appear. No, resistance will occur definitely. It is a biological phenomena, because the bacteria will itself try to overcome the attack of the phages. But so there is a kind of means which wins the race. But it happens that we have seen that if you give a cocktail of phage, then this kind of resistance is quite means up to 100 times it is overcome. So these two things I would suggest, that we should be going for genome sequencing, and we should be going for a cocktail for overcoming any kind of resistance that might come up later. So we should be
Ma'am,
selecting.
In whole genome sequence, what should be we have to like what all the genes should be studied, like virulence genes or integrons? What all the parameters should be followed?
Virulence genes should not be there, like antibiotic resistance gene should not be there. सबसे important, most important is integrase and recombinase should not be there. Then lysogeny related genes should not be there. So these three, even if you suspect some genes like this which are lysogeny related, or which are these ones which I have mentioned, they should be completely avoided first of all.
Ma'am, for stability, what we can do further for the like phases, because we know that these are really like they respond very suddenly, and even though they may get the resistance in future,
That is
to
why
avoid that.
you should make a cocktail of at least 4 to 5, 3 to 4, 5 good broad spectrum phages. Because if it is resistant to one phage, then it might be attacked by the second phage. If it is resistant to the second phage as well, it might be attacked by the third phage. So select broad spectrum phages and make a good cocktail.
Ma'am, this cocktail should be of same host or different host, we can do it.
No, if you are targeting only one host, it should be against one host only. And if you believe that there are different hosts, like for example, you want to target Amibominii as well as Pseudomonas aeruginosa together, then you should be having broad spectrum phages for both of them together. So it depends upon what is your aim actually. But phages
तर
are very specific in action. It does not happen that A. boehmeni phages will also be active against P. aeruginosa. This probably would not happen.
आता
Ma'am,
चालू आहे
actually,
संशोधन.
I am having Acinetobacter baumannii species like different 2 to 3 strains. I have isolated for all the strains different phases, and I have tried one phase on all the strain, like 5 strain I have tried. So all are giving the lytic activity. So I can conclude all phases are same, or based on the strain there
So
may be
5
a
is not
variation.
enough. 5 is not enough. You should try your phages at least on 30-40 host strains. Like A.
I have
boehmeni, different strains, at least 30-40 you should be trying.
tried,
And
ma'am. Actually, I
moreover,
have tried for around 50, but I got only around 5 samples were got for the lysate. So I'm
uh-huh.
focusing on those only. So that's what I'm asking. Like difference between strains can vary according to the phases.
Sorry, I'm not exactly getting your question.
Strains may be different, so that my phase may not work on those all strains, ma'am.
Yeah, it will happen. It will happen. Because the phages are narrow spectrum also, you might be knowing well as you are working that they might be strain specific also, and they might be species specific also. So this will happen. But the basic strategy is you generate a cocktail of the whatever broad range is possible for you, that broad range you select. And you can do EOP studies also, like efficiency of plaques formation study also before that. Okay.
Okay. Thank you.
Welcome.
Tapita has clarified her many doubts, but she has to tell her what exactly work she has done with Dr. Chandrakant Kelmani, because Dr. Chandrakant Kelmani is my very good friend. So she has.
Sir, actually, I have isolated Acinetobacter baumannii from hospital samples, like
No,
around 185 sample I have isolated. Around
no,
we
no.
are, I am
See,
having.
one minute. You see, your host should be first identified one. You can't
Yes,
go
sir.
after
Yes, sir.
isolating host also, isolating phages also. All right.
Yes, sir. I have confirmed with 16S RNA
No,
sequencing.
still okay. Next you can.
Next I have isolated the phases by using hospital effluent of ESIC hospital is there near to our university. So I have collected from their hospital effluent.
Okay.
Then I have gone for enriching the phase with the effluent. Then after 24 hours, I have gone for the checking out the lysate by using spot assay. Then I have gone for serial dilution. Then
So
I have gone for
you checked against the single host or different host of Acinetobacter?
say I have tried around 70, sir, but I got only for 5 strains to get lysate for the phases.
No, your host. Host is a single culture or different cultures?
Host is same, sir. Acinetobacter baumannii only, but
Okay.
strain is different.
Hmm.
And other also I tried Pseudomonas and E. coli, but I didn't got
No, you have to be specific, no?
a specific, sir.
If
But
you
for
want to again the
applications,
Acinetobacter, so you should take the different species of or strain of Acinetobacter, right? And then see which phase actually is infecting. What we have that multiplicity of infection, right? MOI you should actually
yes, sir.
identify. Yeah.
I have done,
And
sir.
then
This
you
MOS
can see
studies
the
are all
host
done.
strains. You know how
Yes, sir. Both
and then
so then I have gone for TEM analysis, like what is the morphology of that phase. Then further, I need to go for whole genome sequence. I'm getting like I have to know what all the genes should be there or not. So further, I am asking for a few of the like institution for which may help for me like whole genome sequencing, etc.
whole genome
Actually, it is
sequencing
Acinetobacter baumannii.
whole
Yes,
genome
sir.
sequencing is not the big issue now. Everyone is doing whole genome sequencing. Several companies
But
are doing it, so you can actually get it done. And as other
correct.
expert also told that your path should be the pure one.
Yes,
Otherwise,
sir, it is
you
pure
can
for sir. It is a pure. I have already isolated. I gone for like 3 to 4 trials for lysate, and I have collected 3 to. I have repeated that like around 4 to 5 times with using single lysate only. And it's my organism is actually extensive drug resistant, sir. Acinetobacter baumannii. It is resistant for all carbapenem as well as ESBL resistant.
Wonderful. Then you have actually a very good chance to come out with the good results.
Yes, sir. I'm getting the results, sir. That's only now I'm in this whole genome sequence studies. And after that, I need to go for isolating the protein. I need to go endolysine or what. So what are the possibilities? So I need to go for further, sir.
Yeah, but that you can't change. No, you can only eliminate from your screener for its further utility. If it has
Yes, sir.
indolysine or any recombinase, if these undesirable genes are there, you can't use that further.
That's what, sir. That's only whole genome sequence. After that, I will plan what may be done, like what are the
Exactly.
genes.
Or otherwise, just to have a basic study is the host, no, host strain, the specificity, all these things you can do it. But for
Yes, sir.
application,
That's
you have
what.
to actually see what are the selection criteria are useful properties it should have. That you have to
Yes,
apply.
sir.
We can
Yes,
go
sir.
to this Karunya GR's question looks quite interesting. Dr. Taruna can answer, I think that. Mycobacteriophage for tuberculosis to combat MDR in TB. That
Yes,
looks
sir.
interesting.
Sir, there is a very dynamic lady, Dr. Urmi Bajpai. She is working on mycobacteriophages for treatment of tuberculosis. And she is the vice president of our society also. And she is right now in Delhi University South Campus. So she would be the best person to contact in this regard. You can definitely contact her. She is very cordial and very means she will definitely respond to you about it. Dr. Urmi Bajpai.
Yes. Similarly, Dr. Neelam Tanuja also. In fact, she is my collaborator actually on AMR. I hope you are referring to Dr. Neelam Tanuja, right?
Yes, yes, sir.
Okay.
Dr. Neelam Taneja, she is in, I told that she is in
PGI
PGI
Chandigarh.
Chandigarh.
Yes, yes.
Yes, yes. She is very nicely working on fast therapy for patients. She is trying
Exactly.
to actually purify
Those,
phages and giving
those
them to
participants
patients.
wants to do the clinical trial, I think they can approach her. So
Yes,
this is a very
definitely,
good
sir.
platform.
She is a very good practitioner. Last time she was there in our conference, sir. Last year. Yes, sir.
Okay, okay. We were actually associated with AMR for preparing National Action Plan for WHO India office.
Yes, sir. Yes, sir.
I think Manohar's question also is interesting actually.
May you please
Kindly
repeat?
share the details whom to contact in case if one wants to learn isolation. Just that madam from Chennai, she told SRM
I,
University.
sir, isolation, I am telling na, this time we are holding a workshop, pre-conference workshop also.
Exactly, yes.
Yes, sir. In from November 1st and 2nd. So that is a 2 days conference, and we are showing all these aspects, and it will be completely hands-on. So for students and even assistant professors and all, it's very good to be. We are showing end to end means starting from phages, how to isolate, to characterize, and to genome and everything we are telling in that workshop.
So
I
Manohar
hope.
can contact madam, I already informed. And then this conference is going to take place already. I'm also seeing the website on ICBRAR 2026.
Yes, sir.
Which is going to take
Yes,
place in November 1 to 2 is a workshop days, and then main conference was on November 3 and 4.
yes, sir. Yes, sir.
Yes, exactly. You have some
And
information
uh,
pertaining
uh,
to
ours
bacteria.
is a complete lab which is established under ICR. It is exclusively on bacteriophages, no other work. Only bacteriophage. It is like one of its kind lab in whole of the ICR. So, and I hope we'll be able to deliver as per expectations of students and researchers who are working on bacteriophages or who anticipate to work on phages.
Exactly, madam. You have some information pertaining to the what you were referring this bacteriophages obtained from Ganga water?
Sir, Ganga water, yes, we already conducted two trainings, and we have published a paper in 2016, 2018 probably, where we isolated a phage from Ganga River, and it was against multi-drug resistant Klebsiella pneumoniae. So what we did was the bacteria was inoculated into mice to develop a model of infection, and later on it was given phage therapy so as to overcome the lung infections, and complete pathological investigations and molecular studies, and they were done, and it was observed that phage therapy worked wonderfully in mice model to means protect and to in means for therapy of lung infections, and the mice were overcome means they were overcoming infection very easily and earlier as compared to the natural process or given when given antibiotics. So in that case we actually isolated phages from Ganga river against MDR bacteria.
Wonderful. I think Ganga water must be the very good source, I think, for isolating different
Yes,
bacteria.
actually nowadays, sir, every river is a good source. Basically, we dispose a lot of waste materials. Our community is like that. They dispose a lot of waste materials into every kind of river, and so these make them a good source of phages. And nowadays it is, you can say that it is not restricted to Ganga. You can find phages in soil, you can find it in sewage, you can find it in even from air it has been isolated, phages have been isolated. Some people tried from Godhuli, they tried. So means, but that spiritual angle is right now not that much associated with the phages. But we obviously we started with because initial studies were uh, designed that way that it initiated with the Ganga River. But now on, we find that phages are present almost everywhere. Where bacteria is present, phages are present.
I think this also question can be interesting. I think anyone working on uses of bacteriophage in agriculture to control bacterial diseases.
Sir,
Taruna ma'am,
in case, ji sir,
hello.
ji sir,
Ma'am, I have a very basic question that in
ji sir.
UG we are giving some I mean, discussion and practicals on isolation of bacteriophage from sewage in semester 5 and 6. So what happened, ma'am? Sometime we did not get plaques on top agar.
Yes,
We are using top agar and
yes,
all, right?
yes, it
So,
can happen.
so
Yes,
ma'am,
it can happen,
ideal.
sir. It can happen.
So
It can happen in two, three cases, sir.
कई बार क्या होता है कि 3 दिन का प्रैक्टिकल होता है, क्लोरोफॉर्म ट्रीटमेंट देते हैं, वो सब कुछ होता
हाँ,
है, देन
हाँ,
बच्चे सुपरनेटर निकालते हैं एंड दे आर यूजिंग तो टी ब्रॉथ में,
हम्म,
ट्रिप्टोन ब्रॉथ में यूज करते हैं, वो अच्छे से प्रोसीजर करते हैं, बट स्टिल
हम्म,
प्लाक्स नहीं आते। उसका रीजन मैम क्या होता है? क्योंकि हम तो क्या बोलते हैं, प्यून को बोलते हैं कि भाई आप गटर का पानी, सुवेज वाटर जो होता है ना, वो अर्ली मॉर्निंग में लेके आ जाओ, कि अर्ली मॉर्निंग का पानी जो होता है सुवेज का, क्यों? तो क्या मतलब रीजन क्या है, क्या कौन सा वाटर लेना चाहिए एक्चुअली इफ यू वांट प्लाक्स,
हम्म।
परफेक्ट प्लाक्स अगर चाहिए आपको, तो क्या मैम को करना पड़ता है इसके लिए? कैन यू गिव
Uh,
सम सजेशन?
Sir, two, three things I would like to suggest. First thing is that if your sample is not giving a phage against a targeted bacteria, my question is like against a, my point is like it should be one strain. There should be no mixture on the plate, that it no bacterial contamination should be present. First is, if you are not able to get a phage against a targeted bacteria, then there are only two things which I believe. Either your targeted strains phage is not present in that sample, because if it is there, it will come. Or you can modify the agar composition. You can dilute the top agar more. Like generally we use 0.7% agar as top
Yes, yes,
agar.
yes.
So it can be diluted more, like 0.4%, 0.5%, like that. That could be
Okay.
the second thing. And the third thing is like, sometimes it happens that phages give too tiny plaque that it is not visible to the eye. So,
Yes.
so we can introduce a dye, that is a TTC dye, that can be incorporated in 1% concentration in the top
Okay.
agar. So it will make the top agar red. So when, even if a tiny plaque is there, na, it will be visible.
Okay. 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 Okay, ma'am.
So,
Yeah, we'll try. Yes. Thank you
these
so
things
much.
can be done. So, anything else, sir?
Ma'am, I have
Yes, Prakash
recently
sir.
isolated a bacteriophage from environmental sample. Could you briefly explain the workflow for isolating the phage genome after purification? Like, also, are there any well-established DNA extraction kit that you will recommend for phage genomic studies?
Sir, there is one DNA extraction kit available for phages, probably Newgen or something, Newgen. But
Norgen,
Norgen, yes, Norgen Biotech, yes, it is there. But there I observed, personally, I felt that it did not work very nicely for me. Maybe I did it wrongly or what, I don't know. But it did not work nicely. We follow our own lab's established protocol for isolation of DNA, and very pretty good DNA in very huge अच्छी concentration में it is obtained. So isolation of DNA of phages is not that much of a problem, because we have a whole genome sequencing in our lab facility. We have developed it through the National Fellow Project, and we are doing all the genome sequencing in our own lab, not sending anywhere else. And we are doing all the bioinformatic analysis also, our students are doing on their own. So genome is coming nicely. Sometimes some tidbit problems they happen, but generally DNA isolation is okay.
And
So
can
you
you
can
share the
try that
protocol?
manual process. Manual process.
And can you share the protocol?
Yeah, it is available in my paper, sir. You can download any paper. PubMed पे से आप paper download कर लेंगे, मिल जाएगा. नहीं मिलेगा तो I will definitely share. No issue.
No, thank you.
Anything else, sir?
Okay. Thank you so much, ma'am.
Thank you.
Really, it
Thank
was a
you.
very nice session. Prakash sir, are you there? Prakash sir?
Anand sir,
Yeah,
Yes.
Dr. Prasun is there. We can take his
yes,
opinion and then we can conclude the session.
today I was listening since the beginning.
Oh. Oh.
Yes,
Please give your inputs.
yes. Just I had a question from to the expert, na, Dr. Dharna ma'am and Dharna ma'am is there. So, uh, one point that I noticed is okay, endotoxin and recombinases should be absent. But how can we know about this? So, is there any test before going for
There is
gene sequencing?
there are kits available. LAL test kits are available, and there is a permissible limit and detection limit for that. LAL test can be used, and but keeping in mind that I would suggest that you should be trying many samples at a time. It is a costly kit, and it expires very soon. Within 5-6 months, it's expired. So there is a LAL test kit. It is available, and we can use it. And for purification, once we detect the limit, how much of endotoxin is present in a sample, it can be like washed off by protocols. There are two basic protocols available, and they can also be shared. Rather, they are very freely available. There is one cassette based microfluidic cassette based purification system also that is available, and even SRM they are using it probably. I have come to know SRM Dr. Govindarajan, Sudarshan Govindarajan, they are using that kit, all that system.
Okay.
But it is established and it is well known, and you will find it easily.
For whole genome sequencing, I think we should go with this test and then go for whole genome.
No, whole genome sequencing is not. We should be going whole genome sequencing first in order
Okay.
to find out whether this phage is useful to be pursued further or not. Because once you establish that a phage is useful to be pursued further, then you invest in purification of that phage.
But if in the whole genome itself we can get the means endotoxin or recombinase gene presence or absence, we can get to know from the whole genome or not.
No, endotoxin gene is not there in the phage.
Okay.
Phage, it is released by the bacterial membrane.
Uh,
When the bacterial membrane degrades, then endotoxin is released. So it has nothing to do with the phage genome.
so mostly gram-negative bacteria will release endotoxins.
Yes,
We have
yes,
to.
yes, yes,
Yes.
correct. Yes.
Okay. And the dye name you said na for a smaller plaque that TTP.
TTC dye. It is tetrazolium, some chloride, something, tetrazolium chloride, something like that.
TPTC. TPTC is
TTC.
a very common dye used for seeing the viability.
Viable, sir, it gives a red color
Yes, yes.
to the medium.
Mhm.
And after adding to it, you can autoclave, and then you can make like top agar, and it will, whenever plaques are there, they will be appearing like on a red background, clear zones. They will be more prominently visible.
Okay. Okay. Thank you so much, ma'am. One last request. Recently, I used it through my BTech student. I isolated few phages. He was very much interested. He wanted to ask if he can go for a training because I am not much expert of this area.
I'm
So,
telling you,
yes,
training we are only holding pre-conference workshop
yes.
November 1st and 2nd. So they can register for that workshop. Very minimally paid, and it will follow with the conference also. So students will get an opportunity to meet many very like Yves Briers is coming, and Robert is coming. Many well-known Faj people are coming because this is a Faj associated society. So only those means which are exclusively and very high-endedly working on Fajes, they are coming.
Yes, yes.
So,
Even I would like to join. I will get permission from my department. I will come. Yes.
okay, okay. Most welcome, most welcome.
Thank you so much, ma'am. Today
Thank
is
you.
was a very nice discussion, Anand sir. It was very useful for me, at least for my personal knowledge. And thank you to other expert as well.
Thank you.
Ma'am, I have a question.
Yes, please.
What advice would you give to MSc microbiology students who want to pursue research or clinical microbiology?
See, clinical microbiology, I would say that first of all, every clinical trial has got different stages. Trial 1, 2, 3 means there are different levels, no? So you have to have a good team so that you need to, whenever you need to assess the various parameters, you are able to do it. First of
Yes,
all,
ma'am.
you need to complete in vitro studies, followed by in vivo in small animal models. And
Yes, ma'am.
do not hesitate because sometimes no, it happens. I have seen that people would take just one mice or two mice or maybe one bird or two bird. Don't do like that. Please take appropriate number of animals which are required to appropriately conduct a study. And then if your study is giving you good results in vivo studies, then you definitely will have to have a medico team along with, and you have to means develop that way your procedure. It has to go. There is a set pattern like trial 1, 2, 3, and then and
Yes,
you
ma'am.
need to assess what all parameters you should be having with you, so that eventually when you report your study, it doesn't go away in vain. So just consult good clinicians and make a good team.
Okay, ma'am. Thank you, ma'am.
Thank you. Sir, I hope I have answered questions appropriately, and still, if there are more questions, you can write to me on my email. No issue, sir. I would be there to help.
Dr. Anand, you can conclude now. Already 9 o'clock. You are what, ICAR National Fellow there?
National Research Center on Equines, and I am National Fellow, sir, right now. National Fellow.
And this is what some sort of a
Sir, this is a competitive grant and competitive position. Actually, many people they compete for this position, and all over India, only 25 people at a time are made national fellow. So it's a competitive grant where you are given a project with main aim and for 5 consecutive years. So it's a kind of position which is given in ICAR to.
this is what 5 plus 5 years.
I will take it for 5 years probably only, not below, not more than that. But it is 5 plus 5 years.
Wonderful.
Yes, sir.
Equinox.
Taruna ma'am, if that brochure is ready, can you send to Prakash sir or in microbiology group? If
Yes,
brochure is
everything
ready,
is
then.
ready. Brochure, website, registration, please do through website. It would be very much convenient for you. And all the participants, they can easily register through website.
Sure.
They have to first make the payment and then a code will be generated. That code will be used to submit the abstracts. So it is very
Okay.
easy. Everything is just lined up. No issues, no hassle.
Okay.
And
Okay. Okay.
registration
Thank you
fees
so
is
much.
also reasonable as such. Though it is an international event, I have
Yes. Yes. Correct.
verified. Otherwise, in most of these conferences, registration fees will be very high.
Sir, actually, we have taken a great care altogether to keep the fees nominal for students at least,
Very
so
good.
that students can learn about phages. They can pursue this as a option for many more things. So just to keep in mind that every time we have to keep fees very nominal for students.
Very good. So your culture collection
Okay.
is only for the bacteria, right? Bacteriophages, not
Yes,
other
sir.
viruses.
We have viruses also, where viruses, bacteria, bacteriophages, clones, cell lines,
Animal
everything.
viruses or plant
Animal viruses, yes, sir.
viruses also.
Veterinary animal viruses. We have, our institute has produced the LSDV vaccine, the LSD vaccine, no, that is produced at our center, sir.
Wonderful.
Yes, sir.
Dr. Anand, you can conclude now.
Yes, sir. Okay. Okay. So today's session really it was very interesting session, Taruna ma'am. It was a very useful information about the fast therapy and
Thank you.
whatever that. Yes, really it was a very fruitful discussion with all the participants. Yeah. And thank you so much, Deshmukh sir. With the very beginning, with the very start, really, tomorrow is Guru Purnima, and today only we got a very good information. about this bacteriophage therapy, and other participants also discuss about this phage therapy, and some MSc graduate students and PhD students also raise some questions. Thank you, Prasoon sir, Viprant sir, and Prakash sir for conducting this type of the topic. And Viprant sir already suggested that we can go with this topic. Today only we discussed topic in morning, and we discussed with him, and with the permission of Deshmukh sir, we continue with this topic today. Next week also, I think if it is possible, Alami sir, we can go with this topic. If it is possible, we'll discuss with him, and then we will.
Yes, sir.
Yeah, this is very interesting topic.
Part 2, that is therapy, has not been covered.
Yes.
We can actually extend to the application part. What I mean.
Application part. Yes, correct, sir. We can go with application. We can just change title, and we will discuss, sir, next week. Yeah. Okay. Thank you so much, everyone, for joining this.
Thank you so much.
Thank you, madam.
Especially Deshmukh sir, I am really thankful, sir. Thank you, sir. Thank you, sir. I'll send the brochure and everything in the group also, so that those interested
Done,
can
ma'am.
register easily.
Exactly.
Yeah.
Thank you.
Great.
Thank you,
Thank you,
everyone.
ma'am.
Bye.
Bye, everyone. Bye, everyone.
Good night. Good night.